This is a production of Cornell University.
Thank you, Ed. Thank you all so much for the chance to be here today. It's been really fun, both the conversations I've had over the last 2 days, and also just getting to wander around campus again after about 15 years since I first showed up here. So we had a conversation at dinner last night about some of the digital ag stuff that is going on here at Cornell. So I want to highlight, before I get into my talk, this is my research group as of last summer at the University of Nebraska-Lincoln. But a lot of the stuff I'm going to tell you about is only possible because we were collaborating with folks like Yufenga, who's a biological systems engineer, and Yumo and Yuhong, who are both statisticians at the University of Nebraska. So it's really being able to work with folks across different disciplines that lets us do really, really cool stuff that to me seems extraordinarily hard, and to them is quite easy. And then we get to turn that around. Also, Brandy Sigmund, who is an actual developmental geneticist, which is fun. So I'm going to start with a sort of broad question. Then we're going to move into some phenomics, then into some quantitative genetics, and come back to sort of broad conceptual stuff. So that broad question here, which is, I think one of the central questions of all genetics, is how do we find which genes determine the characteristics of maize or any organism? And I like to bring this up because I want you to think about what actually we mean when we say the word gene. So when I first started out in grad school, the maize genome had just come out. There were about 32,000 gene models. Then there was a revision that went up from about 32,000 to about 40,000. Another revision, it went up to about 60,000. So about the time that I got my own lab, we were adding about 5,000 genes to the genome every year. Now, if it takes one grad student 5 years to characterize one gene, you can see that just for maize, we would need about 25,000 graduate students and about $1.25 billion a year in funding to keep up. Now, obviously, this would not be sustainable for a lot of reasons, working with government policy and NSF and all that. It also is clearly not what's happening. Maize is not adding a lot of new genes to the genome. What was happening is we were using different criteria and had different evidence for what constitutes a gene model. So if you go into Google Scholar and you type in what is a gene, you will find this review from more than a decade ago now, where they proposed this very complicated and almost legalistic definition of what a gene is. And I will say that, Ed mentioned I've worked with some companies, and there's some very good things about that. One of the negative things is you spend a lot of time talking to lawyers about IP law and organizational law, and who's going to have control of which part of the company if one of the partners gets divorced, especially the partners who aren't married yet. So it feels like a lot of pressure to have to be divorced before you get married. Anyway, so I would say that this is a definition that only a lawyer could be happy with. So if you go back to the origin of the term gene, and I didn't know this until about 6 months ago, it was originally proposed by this Danish geneticist. And this is not the original Danish. This was a translation to German. I know just enough German to find the right sentence to paste into Google Translate. The word gene is completely free of any hypothesis. It only expresses the assured fact that at any rate, many characteristics of the organism are conditioned by special, separable, and therefore self-existent fundamentals that occur in the gametes. So genes are the things in the genome that actually control the characteristics of the organism. And I'm going to argue, not all gene models are going to correspond to genes. So one of the wonderful things about the grasses is that they have very conserved synteny. So synteny is the conservation of the same genes in the same order. So what you're seeing here is a graphic from Koji, which is an online comparative genomics tool. It's run by Eric Lyons at the University of Arizona. And each of these, we have a nice gene model comparing a region in rice, in foxtail millet, in sorghum, and in maize. And these red-orange boxes are regions of homologous sequence identified from BLAST. And what you can see is that we have some of the same genes in the same order. There's also a tandem duplication in sorghum, so we have 2 copies of this particular gene. I think this might be phytochrome B. I'd have to check on that. So we have these genes that are gene models that are conserved at the same locations across all the grasses. We also have lots of gene models that are not conserved at the same locations in different grasses. So these are synthetic orthologous genes. These are non-synthetic genes. And the fact that we see them here but not in other species means that they inserted into their present location in the genome since the divergence of these different species. So if we look at the genes that actually have known characterized functions, so some geneticist walked through a field, they said that plant is so interestingly weird, I'm going to spend those 5 years of my life figuring out what is the gene responsible for why that plant looks weird. Those genes are 8 times as likely to be these conserved synthetic genes as the non-conserved, the non-synthetic genes, which are evolutionarily more recent and may or may not have functions. Now, there's also an argument to be made that maybe these non-synthetic genes are involved in processes that aren't as immediately obvious as you walk through the field. There's at least one exception where that is the case, which is if you look at disease resistance genes, these are almost always come from the non-synthetic fraction of the genome. But a lot of these genes do appear to be sequences with the potential to code for protein, or that actually do code for protein, but they're in search of a functional role still. And there are lots of other ways that you can see differences between the conserved synthetic genes. So they tend to have very different methylation profiles, the synthetically conserved genes. The non-synthetic genes tend to look a lot like intergenic sequence. Synthetic genes are expressed at higher levels. This is from a Wally paper a couple of years ago. But more interesting to me, if you look at non-synthetic genes which are expressed, they make significant amounts of mRNA, they're much less likely to have that mRNA translated into protein than the conserved synthetic genes. So if this would be very consistent with them having fewer phenotypic consequences, because even though there are ways to produce phenotypic consequences without making a protein, it's a lot easier to have a phenotype if you do make a protein. So given a lot of these differences, we put together just a very simple PCA analysis. So what you see here is a 2D. Anyway, you're seeing essentially a kernel density plot of both all the annotated gene models in maize in red here, and these gold standard genes that have been identified based on a forward genetic phenotype. And this is a subset of the trait. We put about a dozen traits into this basic PC based on ones we knew that there were significant differences between genes with known phenotypes and those without. And we see that there's a relatively large separation. So a lot of the annotated genes in the maize genome are concentrated over here. They're more likely to show presence-absence variation. They're less likely to be synthetically conserved. They're less likely to show evidence of being translated into protein. But the genes with known phenotypes tend to cluster over on this side of the distribution. So with this, I'm going to give you a few takeaways from this very broad idea, which is that we really do need to remember that gene models are hypotheses. So a gene model is a hypothesis that this particular portion of the genome is actually a gene. It actually plays a role in determining the phenotype of an organism. And I've been very guilty of this myself, that I sort of treat all gene models as equally likely to cause phenotypes. And that's not true, and we wouldn't treat that any other kind of hypothesis the same way. There are some hypotheses that are more likely to be true, some that are less likely to be true. The ones that seem very unexpected but then end up being consistent with observation are really, really exciting. But we can do a lot to prioritize which hypotheses it makes sense to test, which hypotheses you spend a lot of time studying. I think we can do a lot better job of this with gene models as well. I'm going to come back to that at the very end of the talk. But now to jump a little bit from very broad, how do we identify the genes likely to have any phenotypic consequence, to linking specific genes to specific phenotypes. Now, it was funny. I was meeting with some folks at the lab yesterday, and they had reconstructed almost this exact same slide. I think probably everyone working in high-throughput phenotyping has a version of this, which is that genotyping has gone from extraordinarily expensive to essentially a trivial part of the cost. Honestly, collecting the samples and doing the DNA extractions may cost more at this point when you factor in the labor. Phenotyping, on the other hand, has not gotten substantially cheaper. If anything, it's probably gone up because the cost of human labor, particularly skilled human labor, is going up faster than inflation. High-throughput phenotyping is a reaction to the recognition of this imbalance between the decreasing cost of genotyping and the constant or increasing cost of phenotyping. It's also an interesting concept because there's not a good definition of high-throughput phenotyping. It's defined by what it's not. So high-throughput phenotyping is not driving a combine through the field and getting yield. And it is not sending a bunch of students into the field with protractors and rulers and notebooks to write things down. Because the students who are willing to do that, or willing to go out in a cornfield or a sorghum field in the middle of the summer here in Ithaca, Nebraska, they're a very limited pool. I don't think we can really grow that pool very much. They're a non-renewable resource. So if we want to get more data, we need other strategies. But as long as you're not in either of those two things, you can say that you're high-throughput phenotyping, as long as you're measuring something about plants. So it's all right. Now we want to invest a lot in high-throughput phenotyping, but it's really not clear what it is. So I'm going to walk you through the three major steps or three major bottlenecks we found in terms of actually using this to do something useful to understand how plants work. So we have a little flow chart here, and there are three transitions. So growing plants, I do know how to do that. I'm not great at it. I kill them occasionally, but we can grow plants, and we can collect traits from them. But the first challenge is an engineering challenge, which is once you have all these plants, how do you collect new and interesting kinds of data from them other than having somebody out there with a notebook? And so this is an engineering challenge. I mentioned I'm fortunate to work with this engineer, Yifeng Gott, University of Nebraska. So we both have this field-based system. Frank is a former postdoc in Yifeng's lab who's now on staff just to keep this field-based system up and running, where we have a large payload that can move back and forth throughout the field. And we can send up both heavier and more expensive instruments on this than we could on a drone, because we don't have to worry about it crashing or veering off or anything like that. The other system, which came online about the same time I was hired at the University of Nebraska, is just one of these conveyor belt greenhouses. Some of you may have visited Danforth or Pioneer, so you've seen these. The thing that sets Nebraska's apart from other people's is not that we can grow more plants, not that we're cheaper, it's really expensive to run, but the fact that we can grow plants to such a large size. So with this facility, we can grow plants up to about 2.3 meters and still move them through here and have them imaged with a series of cameras every day, collect weights and the specific amount of water being used by each plant in each day. So that's enough to grow grain-type sorghums all the way to maturity. It's enough to grow some, but not all, maize inbreds to maturity. If you try and grow maize hybrids on this, they get above that 2.3 meter threshold and start hitting into the ceiling as they go through, and you have plants toppling over, and that's not very much fun. And then it turns out the greenhouse staff have to detassel your plants so they can still move through the system, at which point I would argue there's not a lot of useful quantitative data. But what you get out of this is a series of different imaging types. So what you see here is one plant being imaged on one day. So we have an RGB camera. The plant is photographed from different angles around about 180-degree arc. We also have a thermal IR camera. You'll notice that the plant is about the same color as the background. That's because originally these plants sat in the dark for about 10 minutes before they were imaged, at which point they equilibrate with air temperature. We've now fixed this by having a tunnel where they're under high light and exposed to the same heat and air conditions as in the greenhouse before they're imaged. Fluorescence camera, which I'm not going to talk about very much today, and a hyperspectral camera, which I am. So as you know, in the human eye, we have 3 different cones to perceive different wavelengths of light. The hyperspectral camera, in this case, is perceiving 243 distinct wavelengths of light. So there are lots of colors that are going to look the same to your eye or mine that can be distinguished with this camera. So we have lots and lots of data, and I'm working with great engineers to collect the data. But there's a step that I think is underappreciated. Probably you folks do appreciate this, but when you talk to people outside of the plant biology community, it's very hard to do QTL mapping or GWAS or anything on a bunch of pictures. I've actually seen people propose ways to do that a little bit, but really what we need is a giant spreadsheet of numerical measurements of something we care about with the plant. And that becomes a computer vision or a machine learning challenge. How do we go with pictures or LiDAR point clouds or hyperspectral data cubes and actually measure things we care about with the plant? And there are a lot of approaches to that, but I'm going to tell you about a couple things going on in my lab that I think are pretty interesting. So one approach is to use neural nets. So you give the computer lots and lots of pictures with known traits, and it will learn from that how to score new pictures. The challenge we have is that even though we have all these high-throughput phenotyping facilities, actually getting enough training data to train a neural net can be challenging. So this is work actually by an undergraduate in my lab, Tom Hoban, who took software that was originally designed to make the 3D models of plants for video games and used this to create models of corn plants, which he could insert into a semi-accurate replication of the same chamber we were collecting real plants from. So in a matter of about 8 hours, he could make about 26,000 of these synthetic corn plants with different parameters and known 3D structures. We worked out what it would have cost to grow these on the actual phenotyping facility. It was about $80,000 worth of data. And we can use these simulated images to train a computer to predict real images. What you're looking at here is prediction of leaf counting. So this is the correlation. If you just show pictures to 2 different people and have them count the number of leaves on the corn plant, the correlation is about 0.9. So it's good, but it's not perfect. That's the first thing to recognize is we're not going to get a perfect correlation because we can't get perfect correlation between different human beings. So our goal is 0.9. We're not able to get there. What this is showing, and I won't walk you through in too much detail, is that when data is very scarce, so about 300 images per class or less, adding in the synthetic data actually improves our accuracy over just using the real images. And these are not particularly accurate representations of a corn plant. I mean, I hope it's recognizably a corn plant. There's clearly a lot more we could do to make this look more like a corn plant, and so increase the usefulness of simulated data. So that's one approach to teaching computers how to measure things from images. Another is those taking advantage of things like hyperspectral cameras, which perceive the world in very different ways than the way a human being does. So what you see here is a hyperspectral data cube from a particular sorghum plant. This is work from Alejandro Pages, who is a computer science undergraduate in the lab. And so what he did is he took a bunch of these images, put them up on a web tool called Zooniverse, which is a citizen science tool. So a lot of this, if you go on the website, it's like game cameras in Africa, and how are there zebras in the images? How many zebras? That sort of thing. But we put up all these sorghum plants and just asked people to say, okay, where are the stems? Where are the leaves? Where are the panicles? And miraculously, some people were willing to do that, even given the option of looking at cool zebras and giraffes. So we very rapidly got a training data set of about 8,000 data points, each of which is 1 pixel. Within each pixel, we have 243 different wavelength intensities. Looking at that, we can develop very separate reflectance parameters for what these different classes of pixels look like: the leaf, the stalk, the panicle, the background. Now, as a human being, it's very easy to look at a standard picture of a sorghum plant and tell me where is the leaf, where is the stem, where is the panicle. It turned out to be a very challenging problem for the computer to do that from the RGB image. But with the hyperspectral data, the computer can do a very good job of separating these different classes. One of the things I think is actually cool is it does make mistakes, and you can see what it's doing is it thinks that the midribs of the sorghum leaves are stem. So you have this dense lignified tissue, and that's getting misclassified. So even the mistakes are things that make sense. I'm going to come back to that. Oh, so the nice thing about being able to separate out the different parts of the sorghum plant is we can start to measure traits that make more sense biologically. So one of the first things whenever our students join the lab is they learn how to segment out this is an RGB photos of sorghum plants, and this is actually the same sorghum plant over time. And the image is segmented, so we have the plant pixels circled in red. You can see there's some mistakes down here. But it's possible to measure the whole height of the plant. What you will note is that the height of the whole plant goes up and then goes down because as the flag leaf first comes up, it's pointing straight up, and then as the panicle starts to emerge, the angle of that leaf changes. So we get this sort of bump in the analysis. By having this organ-aware segmentation, we can instead look at the same sorghum plant and look at just the height of the stem and panicle. And so we get a much more consistent growth curve that is doing a better job of reflecting how the plant is growing than looking at the total height, which is influenced just by this leaf sort of flapping up and down as the plant grows. And this is important because when you look at a lot of traits that are measured from hyperspectral or from high-throughput phenotyping data, you'll get something like this. So these are 2 traits, just plant height and biomass. And here we've got ground truth, which was measured, again, by students with rulers and notebooks and scales. And this is the estimated measurements for both of these from the images. And these correlations look pretty good. This question that my student Jakai thought to ask, though, is the error that's left here, is it random? If it's random error, we'll just throw more plants at it. We'll get really good accurate estimates. Or is it systematic? And what he found was that it was systematic. So there were certain genotypes that were systematically having their biomass underpredicted, and others that were systematically having their biomass overpredicted. So about half of that variation is heritable. It's controlled by genetics. So there are differences in the architecture of the plant that are producing systematic errors in our measurements. So if we were using this to do, say, train a genomic prediction model for biomass, we wouldn't just be predicting biomass, we'd also be predicting the phenotypes that lead to errors in measurement of biomass from high-throughput phenotyping data. So it is important if we can get, getting more accuracy is very important, not just because it's more accuracy, but because we're removing systematic biases in the way that the data is being collected. So now to the fun bit. So we've gone from growing plants. We've developed robots and sensors and things to collect data, and then computer algorithms to turn all that data into giant spreadsheets full of numbers. But I really am a biologist at heart. I would like to actually, map some genes or find something. And there are some interesting quantitative genetics challenges, opportunities, whatever you want to call them, that occur because of the types of data that are produced by high-throughput phenotyping. So this is the same sorghum plant from the last section. You can see the two different measurements of height. You can also see the heritability of height as measured by these two methods, both from the individual data points and also from fitting a non-parametric curve to each of the heights. And you can see that generally the heritability from the fitted curve is a little bit higher than from the raw data. It's not a perfect pattern. And we think this is because we're able to borrow data from adjacent data points to get a more accurate measurement. So another student in my lab, Chen-Yong Yao, is working on using this to do mapping in the sorghum association panel, which is a population of about 400 sorghum lines, most of which are conversion lines. So they were collected from all over the world, crossed to US germplasm to integress both photoperiod and sensitivity genes that will actually flower if we grow them in the summer in Nebraska, and also dwarfing genes so that they will be plants like this and not plants taller than the ceiling. Now, if you grow this population in the field and you're interested in plant height, it's very easy to map at least two, maybe three of the large effect genes that are involved in sorghum height, because there are still some very, very tall sorghum plants in our population. Now, I mentioned in the greenhouse, we can only grow plants to about 2.3 meters. So if we take out the extremely tall plants that are producing a strong signal and use the same data, we now are not able to map any of these dwarfing genes. We know they're real, we know where they are, we know we can't find them with the population we're going to work with. So what you see here are the height curves of a large number of the different sorghum plants collected from this experiment in the greenhouse. So this is days after planting. This is the height in centimeters. And you can see that when the plant gets ready to have the panicle emerge, you have this large bolting response, which happens at different times for different plants. And it just looks very, very messy, and it's not quite clear what to do with it. This is actually pearl millet, not sorghum, because it looks cooler. But you're seeing images, I think these were collected every 2 days of this pearl millet plant throughout its life cycle. As a human being, it's very easy to look at this plant and see, oh, look, there's this transition. It goes from just making leaves. So now it has some panicles up the top, and then it goes into senescence. You will also notice that there's this person who comes in and was doing some sort of maintenance during one of the days while we were collecting images. So again, this is a thing that is easy for a human to do, but Chen-Yong was able to train a neural net using less than 1,000 images to get relatively good accuracy at identifying when is this transition point from reproductive development to from vegetative development to reproductive development. And what's cool about this, again, is that even the errors are the kinds of errors a human might make. So the transition point we're targeting is when does the panicle first become visible. So the computer, there's a little bit of panicle shooting out here. The computer misclassified this as not flowering. This plant is about to flower. The panicle is right here in the stem. You've got the flag leaf up here, and the computer got confused and decided this was a flowering plant. So these are a lot of the same kinds of mistakes that I would expect, particularly a bored human being to make if I was, hiring high school students or people online or something to do these classifications. So once we have this classification data, instead of using planting as our benchmark for comparing everything, we can shift the data around to days before or after panicle emergence, and we start to get much more similar shapes of growth. what you'll notice is now we have different time ranges measured for each plant. And with this, the first thing we can do is just scan through and do a genome-wide association study for height on every different day. Let's see. Okay. So this is, you can see dwarf 2 and dwarf 1, dwarf 2 and dwarf 4, which I think is not actually dwarf 4. I think that's mislabeled. Show up, and then later on in development, we don't catch anything. So that's animated. It's a little hard to follow. So here's another way of displaying the same data. So what you see here on the y-axis is the 10 chromosomes of sorghum. And going across, we've highlighted genomic bins when there's at least one significant SNP in that day of the analysis. And there are several relatively long peaks that correspond to some of the genes that we would expect to be able to map, either if we benchmark on days after planting or if we benchmark on days after panicle emergence. But we're running lots and lots of GWAS here, and if I tell you where the known genes are, we can find them, but it's hard for me to look at this and say with confidence, okay, we really have identified what we want to identify, because there's a lot of noise also there in the analysis. So this is where we started talking to another statistician at the University of Nebraska, Yuhang Xu, who is a recent hire, and his expertise is on working with non-parametric regression and the curves you can generate from that, and actually being able to, how do you break that down into how different individuals vary with respect to those curves. So you see here in gray are all the curves for all of the sorghum plants in our population, as well as both the empirical estimate of sort of the center value or the average pattern and the estimated pattern from his non-parametric regression. And he can then do a functional and principal component analysis to identify what are the major components of variation, which is what you see on the other side of this slide. So PC1, which explains about 2/3 of the variance, is essentially just taller versus shorter. So I never really understand what determines what's plus and what's minus, but you can see if you have a more negative value for PC1, the plant is taller essentially throughout development. You have a more positive value, it's shorter. PC2, on the other hand, is essentially a trade-off. So you have, and this explains another, I think 20 to 30%, I'm going to have it on a later slide, where you're taller earlier and shorter later. So now for every plant, we can describe this curve as a combination of 4 principal components. These two explain most of the variation. And we can do GWAS just using these principal components that describe the shape of the curve as our metrics instead. And when we do that, we get some very clean signals. So, okay, 68% and 28%. Dwarf 1 and dwarf 2 are both identified with principal component 1, and dwarf 3 with principal component 2. And what's interesting about this is we know a little bit about the biology of how these different genes affect plant height. So dwarf 1, dwarf 2 affect essentially all the internodes. Dwarf 3 is affecting the lower internodes, but not the elongation of the peduncle, so the stem that leads up to the panicle. So what you'd expect here is this would have substantial effect on height before flowering, but then you wouldn't. Again, I read them again backwards. Anyway, I'm not going to dig myself a deeper hole in this slide. What I'm going to say is that they affect height differently, and it's consistent with the patterns of the PCs, and I will defer to Chen Yong, my student, on why that all makes sense. All right. So we can integrate data across multiple time points, and we're able to recover statistical power to identify genes that we know are there because we can identify them in a larger population by integrating all this time series data. People have done this previously using parametric models where you decide, okay, plants are going to grow and show a logarithmic pattern of growth. What we found here was not actually that different from logarithmic patterns, but what I like about it is it doesn't force that assumption. So a lot of the really interesting stuff in biology is the really weird, unexpected stuff, and non-parametric curve fitting can capture weird, unexpected stuff, whereas fitting the sigmoidal curves cannot. So the last quantitative genetics point I want to talk about is the challenge of high dimensional or nearly high dimensional data. So technically, I'm told by my statistician collaborators, I cannot call it high dimensional until I've measured more phenotypes than the number of individuals in my population, which is a high bar. So here I'm going to talk about having hundreds of phenotypes, whether or not it is a truly high dimensional data set. Now, because of both high throughput phenotyping and large collaborative projects, it's becoming more and more common to have many, many traits scored for the same population in plant biology. We also see this from medical data record mining in humans, and also from large numbers of molecular phenotypes, so looking at hundreds of metabolites or thousands of gene expression levels across whole populations. And so this presents a challenge of multiple testing, because the conventional approach to doing this would be to do separate GWAS for every single trait. And this becomes combinatorially, or not combinatorial, becomes a lot of tests very, very fast. So a student in my lab, Chikai Liang, working with a statistician at Iowa State, was interested in trying some other methods to deal with this data. And the data set we used was from here at Cornell with the maize 282 association panel. And so you can see that there were large numbers of traits measured, and many of those traits were scored in up to 16 different environments. So we had a total of 260 trait data sets to work with. And the challenge of this is, so if we do GWAS for any one trait, so what you're seeing here is GWAS for a single SNP in a single gene. And instead of having all the SNPs on the x-axis, each of these represents the significance for one of the traits in the data set. So if I run a GWAS for one of these are flowering time traits. A lot of them are statistically significant on their own. If we take into account all the hypotheses that are being tested across all of the phenotypes, none of them are significant anymore. So we would like to be able to do some sort of a more integrated test to utilize the data from all these traits together, because clearly, if you're detecting the signal in many, many different measurements of the phenotype, we should be able to sort of combine and leverage that signal. See, the approach used here is first to identify sets of SNPs within a gene, to cluster and then select a single representative SNP for groups of markers that are in high LD with each other. The first half of this model is just looking at multiple SNPs, but a single phenotype, and is fitting a very complicated statistical model, which I will be upfront is essentially a black box to me. So I have to trust my statistical collaborator and then test that the results that come out make sense. Go through a stepwise selection procedure to select subsets of all the phenotypes that show a significant signal for that gene. Do a new comparison between our initial model, which is attempting to explain the variation for all the SNPs within that gene that we've kept after filtering, using both principal components of population structure variation, as well as a selected subset of the traits from the whole 260 set. I'll say so there's a preprint that will go into the methodology here in a lot more detail. We also have the R code that was used up on GitHub. Because of this stepwise selection procedure, there are essentially really large numbers of hypotheses that are being tested. So the p-value distribution we see is quite skewed relative to what we would expect from the number of tests we're actually running instead of implicitly running. So we end up doing a lot of permutation testing just to determine what is the distribution of p-values we would expect to see, because they end up being very extreme anyway. And from this, we can establish what we hope is an accurate false discovery rate cutoff. And doing this takes quite a bit of time. We had to even making some compromises. So there are things we know we could be doing better that we're not, because it still takes about 7 years of CPU time to analyze 32,000 gene models in maize. And then we get results. And the results look like this. So I already talked you through on the top. We have each phenotype stored here along the x-axis. On the top, you have the p-values for the individual phenotypes. On the second side, you have either the selected phenotypes or the non-selected phenotypes for this gene, and then an overall significance score for the gene as a whole, whether we see a significant association between the SNPs in this gene and all the phenotypes measured for this population. As I think probably came across in the way I explained this, a lot of the stats I don't understand the details of, so I want to see whether or not the associations that are identified by this model make any sense at all. Fortunately, we have a couple of ways to do that. So the first is looking at overlap between the genes that are identified in this population with this set of data in this method, and genes that were identified in different populations using different methods. So here we're comparing the genes that were identified by a number of different models in the 282 population, two genes that were identified in the nested association mapping population, which shares some of the same haplotypes, but is a larger population with different structure. Now, I talked to Ed yesterday, and there may be some problems with this analysis, so we're going to work on some other ways to validate it. But in principle, anyway, if we have more accuracy, we should see more overlap between the genes we identify in our population and the genes identified in a different population for some of the same phenotypes than the genes identified by conventional algorithms. So you see here, we have some data on version 2 of the maize genome and some on version 4, so we're constrained to only the genes that actually have one-to-one correspondence between the two versions of the genome. About 14% of those genes were identified as linked to phenotypes in the nested association mapping population. Then we can look at the subsets of genes identified by different GWAS algorithms, and they are enriched. So they're more like the genes identified in the association, the 282 association population, are more likely to also have been identified in the nested association mapping population. So that's good. That's what we expected. And then this genome-wide, phenome-wide analysis that my student Jakai came up with, we see a substantially higher overlap that is significantly higher than the overlaps seen with any of the conventional GWAS methods. This is an internal control here, so this is multiple SNP, single trait. So it is utilizing multiple markers within the gene, but not trying to do the selection and picking a subset of traits to test together. We can also look at the kinds of genes that are being identified by the Jakai's GPWAS model compared to conventional GWAS. What you're seeing here is a Go enrichment analysis. So when identifying essentially the same number of genes, we see much more enrichment of a wide range of Go term enrichment and purification of Go terms in the GPWAS gene set relative to genes identified by GWAS on the same underlying phenotypic and genotypic data. So I'm not going to go into this too much, but I'm just going to assert that this demonstrates that we at least are identifying a less random subset of the genome, which could be a good or a bad thing. We can also look at the specific phenotypes that are being identified as associated with individual genes that have been characterized through forward genetics. So this is an example of antherior1, which is a classical mutant that was cloned back in, I think, 95. So we know a lot about phenotypes that the mutants of this gene exhibit. We also know the phenotypes that were identified as associated with it based on the GPWAS model. And a lot of these match up. Not all of them, but a lot. So that gives us some sort of qualitative degree of confidence. The thing that I think is most exciting about the genes identified by this method, though, is not any of these, but the ways that they are different from the average gene in the genome. So we're going to compare. Here we have all the genes, our conventional GWAS, GPWAS genes, and another control, which are a set of these classical mutants, which again are those genes that somebody walked through a field and said, that phenotype is so interesting, I need to clone it. So you can see that if we just look at it having an average expression level higher than a single FPKM, all of these sets are much more likely to be expressed at levels above background than the overall set of gene models in the genome. If we look at KAKS, so a measure of how effectively purifying selection is operating on these, we can only do this for the subset of genes that are actually conserved in sorghum. We see that both the classical mutants and the GPWAS genes have a statistically significant shifted distribution towards lower KAKS ratios, which indicates they're subject to stronger purifying selection than the average gene in the genome. There are a couple of other markers. I told you about synthetic conservation, which is almost all of the classical mutants are these synthetically conserved genes. In this particular set, about 94 of them. We also see a substantial enrichment both for genes identified by conventional GWAS and by GPWAS relative to the background set. And conversely, in maize, there are a lot of genes that exhibit presence-absence variation. So they're in B73, say, but they're absent from other genetic backgrounds. And we see that while this is quite common for the gene set as a whole, it is much rarer for genes identified through quantitative genetics. And GPWAS is closer to the really low level that we see for classical mutants. So any quantitative genetics approach to identifying genes that are associated with phenotype identifies genes that look more like classical mutants than the overall population of gene models. And the approach that Jakai is using seems to identify a population that looks even more like those classical mutants than conventional GWAS. So to summarize, we're identifying this population of genes that we think are associated with phenotypic variation. They're more broadly expressed. They're more conserved at the same positions across species. Less likely to exhibit presence-absence variation. They also tend to be longer genes, which we see also for the classical mutants. And so we can take this figure that I showed you at the beginning of the presentation, and we can calculate essentially a gene function score, which is just the inverse of that principal component 1. So a score over here means you look more like a classical mutant. A score over here means you look more like the gene models that don't look like a classical mutant. You can see the distribution of all the gene models in the genome has a large peak over here. And both the classical mutants and the GPWAS genes exhibit similar distributions shifted towards this end of the spectrum. So at the beginning of this talk, I told you that I think we can do a much better job of deciding which gene models represent hypotheses that likely are valid, that they likely do explain something about how the genome determines the characteristics of an organism, and which gene models are much less likely to play a significant role in determining phenotype as a whole. And while I have some students who are working on doing much better jobs of this prediction than what I have right now, I hope this has at least convinced you that such a thing is possible, that we really can look at gene models and do a better job of saying, you know, these probably are interesting and worthy of a lot more study. These probably we don't want to burn out grad students trying to find a phenotype on. So with that, I would like to thank my wonderful lab, my colleagues, both engineering, statistics, and biology, and all of you for your attention here today. I'd be happy to take any questions.
I'll start off with
Okay.
one. How was the bias, or is there a bias for cloning genes? They've already been cloned in rice, or something, or in other species out there, or did you carefully try to get ones that were coming directly out of our maize community or something?
That's a very good question. So that, I mean, I don't have a perfect answer. I would say because we're using primarily genes that were identified prior to 2011, okay, I think the benefit of being cloned in rice was much smaller because a lot of that happened before we had a maize genome to compare to. It would be really interesting. I've got a student working on getting the 2011 to 2019 genes. I would predict we'd see a bigger effect of already having a characterized function in rice or another grass in that later set.
So you could essentially just add that in the model too. You know, when cloned in rice.
Yes. Yeah.
And then.
Yes.
So in the last slide, so I worked on a project during a PhD where I found that genes that are highly expressed in Poaceae species, so we did comparative analysis across 4 Poaceae species, found that genes that are highly expressed also tend to be broadly expressed, and that KAKS is low. So in this model, which tries to predict which gene models are more better hypotheses. Have you tried to tease apart the effects of these correlated characters?
That's a very good question. Let me go back to this. Yeah. So the question was that you've observed a lot of correlation already between the average level of expression, the breadth of expression across many different tissues, the KAKS, which was not in this particular model because we only had it for synthetically conserved genes, and there was another one.
expression level, breadth of
Yeah.
expression, and KK.
Oh, okay. So we certainly see, I mean, that's one of the things I like about being able to visualize the data this way is we do see a lot of these do seem to show up similarly when we project them into the PCA space. I think that there are some genes, though, that we see that do have very high expression but are not evolutionarily conserved. And so being able to pull out some of the categories that are exceptions to the overall trend and look at those would be very interesting in terms of figuring out whether the reason a lot of these are showing up is that they're all correlated, say, with expression, or whether they have separate predictive power. I haven't done that yet. I would be interested to find out the answer and share it with you.
So for the non-parametric regression models, how sensitive are those methods to sparse data?
So that's a good question. So we can configure how flexible it's going to be with individual data points. One of the things I really like about it, though, is it does tolerate uneven data. So if we have dense sampling in some parts and much less dense in other parts, we're still able to fit reasonably good curves. One of the points I didn't talk about a lot with this is the way that the images are collected. We actually can grow twice as many plants as can be photographed in a day. So the only reason we're able to make this comparison is because we're able to use the non-parametric regression to fill in the values so that we can look at the whole population at the same time. Yes.
As far as the counting leaves of the sorghum plants and simulating the images, what's the difference between making up a computer-generated corn and then just taking more pictures of the same, or sorry, more pictures of the same plants you have? Because it won't both lead to some I mean, maybe counting leaves is simple enough for not overfitting, but I was like, why wouldn't you just take more pictures of the same plants you already have to put your training model? As far as instead of simulating images.
So you're talking about just taking photos from more angles, or?
Yeah, I don't know. I mean, like pressing the button more often.
Yeah.
Or,
I mean, yeah.
I know.
Yeah. No, it's a good question.
The simulated plant seems odd to use as a training model. I mean, obviously it helped train the model, but I'm just curious what that difference would be.
So there are a number of data augmentation approaches you can use with real images. You can mirror the image and do adding noise. I think that we would rapidly hit a point of diminishing returns in terms of the number of different angles you can photograph the plant from and still get noticeably different images. So what I like about the simulated approach, and I haven't shown that this is the case, is that in principle we're sampling a really wide parameter space in terms of what a corn plant looks like. And in some ways that's a problem. So I'm not going to jump all the way back to that slide, but one of the issues we had is we don't actually have an alternating phyllotaxy in the simulated plants. The guy who made the corn plant model for video games, I don't think realized that was the thing corn plants did. So we could do more work to converge on a biologically informed 3D model. The bigger challenge, and it's not such an issue for leaf counting, is you also you don't just need the photos, you need the ground truth. And so for a lot of sort of complex structural traits, it can be very challenging to collect the ground truth from large numbers of real plants. If you have the full 3D model of the corn plant, you already know all the ground truth. So it's not just the collecting the images, but collecting the ground truth where you save effort. Yeah.
So the gold standard base phenotypes, how many genes are they? What define gold standard?
So there are about 100 genes, and so these are ones that were identified first by phenotype, and then somebody actually cloned it that way, rather than either just a statistical association between the gene and phenotype, or reverse genetics if somebody said, hey, this gene looks interesting, let's knock it out, and then try really hard to find a phenotype. Yeah.
You've been concentrating a lot on the morphological phenotypes?
Yes.
And maybe much of this then pertains to those because historically that's what people mapped and those are the genes that were cloned first. But what about when you start looking at physiological traits and things that aren't morphologically observable in this way? Wouldn't that shift the entire gene space that you're working with?
That is a good question. So as I mentioned, certainly for the disease resistance genes, those behave so differently from everything else. In terms of the stuff involved in physiology or biochemistry that isn't reflected in morphology, I'm not sure I could make a good prediction right now, but the great thing is we're getting those data sets, more and more every day. So very soon, I'm confident I would have an answer on whether they would behave the same as the genes controlling morphological traits, or if they show very different patterns.
So if I can do a side, Susan, we've done high throughput stuff on metabolites with Mark's Fitch group.
Yeah.
These are big genes in central metabolism, highly conserved, proteases, things like that. They're still in that same axis, as the sediment.
I guess I'm trying to think about the transcription factors that do such different things in different species. And I mean, my take home is a lot of what is regulated, at least the things that we've been interested in, are regulated by transcription factors, and they're A, expressed often at very low levels, and B, very specific times in tissues, and C, they affect the kinds of phenotypes we're interested in, but the same transcription factor does completely opposite things or different things in different species. So that's why I'm just trying to
No, I think it's a
reach into the gene space that I'm interested in, that I don't know if it's collectively.
I would agree that it's not answered, but the data sets are out there, so I need to get you an answer.
Thank you.
Quick question.
Yeah.
I really like your models that separate leaves from stalks from panicles. Those results look really nice. I'm wondering whether the weights for those models as is generalize well to other grasses that you have similar data sets for?
I'm actually, that's one of the things on Shinyoung's to-do list, is to run this. We have pearl millet and maize data and see whether it's translatable or if we need a new training set for each species that we work on. They do work very well across the 400 sorghum genotypes. So at least it's not genotype specific, which is always the fear with a lot of these.
Thanks.
Okay, well, why don't we wrap it up here? Thanks for staying more time.
This has been a production of Cornell University, on the web at cornell.edu.